Centrosomes consist of a pair of centrioles surrounded by pericentriolar material, or PCM. Once considered to be an amorphous cloud of proteins, the PCM is now known to be a highly ordered structure, whose size and organization changes over the course of the cell cycle in order to modulate the centrosome's microtubule organizing activity. Nasser Rusan from the National Heart, Lung and Blood Institute in Bethesda, Maryland, explains that the scaffold protein centrosomin is a key component of the PCM.
Centrosomin is playing a major role in organizing a variety of other proteins, including the final product, which is the gamma-tubulin ring complex. And so we started thinking about centrosomin as it transitions throughout the cell cycle, and Dorothy, with her background in fly embryos, was interested in how centrosomin was organized and regulated in embryos.
In early Drosophila embryos, nuclei undergo rapid cycles of duplication and division without undergoing cytokinesis. Centrosomes remain active throughout these abbreviated cycles so that they can help organize the resulting syncytium. Previous studies have shown that centrosomin forms a compact structure in mitosis that then expands during interphase to form radial fibers known as centrosomin flares. First author Dorothy Lerit was able to visualize these cycles of centrosomin reorganization in live fly embryos.
And by increasing the resolution to acquire several frames per second, you could really see the ball of centrosomin extending out these arms and flaring out and really increasing the total area of the centrosome.
The PCM as a whole expands as embryos transition from mitosis into interphase, but super-resolution structured illumination microscopy revealed that the centrosomin flares extend even further, often reaching around 1.5 microns in length. Lerit et al. Looked for other proteins that localize to this interphase flare zone and discovered that, in addition to its known localization at the centrioles, large amounts of pericentrin-like protein, or PLP, accumulated at the tips of centrosomin flares.
So we made a GFP-tagged full-length PLP construct, and when we looked at it by live cell imaging, we could see these extra structures of PLP radiating out well beyond the centriole. So these are the structures that we refer to as PLP satellites, and we saw them with every interphase. And then as the nuclear cycle progresses and centrosomes enter late mitosis, those structures are basically gone. But then as the centrosomes duplicate and segregate, those structures reemerge, and we saw that pattern faithfully with every nuclear cycle.
Simultaneously imaging PLP and centrosomin revealed that both proteins were enriched at the flare tips.
And we could see the release of these cytoplasmic centrosomin particles, which had been previously described. But we could also detect PLP in these cytoplasmic particles being released from the tips of interphase centrosome flares into the cytosol. And then, perhaps even more interestingly, we could even observe some of these cytoplasmic particles reassociating with proximal centrosomes. So these components are packaged together, they're released into the cytoplasm, and they seem to go back and forth on and off the centrosomes.
In the absence of PLP, the PCM was disorganized, and centrosomin formed longer interphase flares that released even more centrosomin-containing particles into the cytoplasm.
And so if we take a snapshot just with fixed imaging, you can count a huge number of cytoplasmic particles in interphase. And these particles remain in the cytoplasm, and really importantly, we've shown that these particles can organize and nucleate microtubules.
Microtubules were not organized into radial arrays in PLP-deficient embryos, causing defects in centrosome separation and nuclear spacing. This, in turn, resulted in chromosome missegregation, DNA damage, and, ultimately, embryonic lethality. Centrosomin-deficient embryos show a similar phenotype. suggesting that PLP and centrosomin work together to control the structure and activity of interphase centrosomes.
So we want to show if they directly interact and directly form a complex to regulate each other. And so we performed a used-to hybrid analysis with different fragments of PLP and centrosomin, and we show that they interact in two different locations.
Lerit et al. Examined the consequences of disrupting the first interaction site by replacing endogenous PLP with a mutant version lacking the protein's C-terminal domain. This truncation mutant localized to centrioles and to the satellite structures at the tips of the centrosomin flares. However, these structures were disorganized and extended further from the centrioles. Moreover, they persisted throughout mitosis, suggesting that the interaction between PLP's C-terminus and centrosomin's N-terminus helps organize and compact the centrosomal flare zone. Lerit et al. Then examined a previously identified centrosomin mutant, which, they discovered, disrupted the protein's second PLP interaction site.
And we were quite surprised to see that while PLP localized to the centrioles as it normally would, what was missing were these extra structures, the PLP satellites. So the centrosomin B4 mutants no longer form these PLP satellite structures, and they show this massive disruption to the PCM structure, similar to our PLP mutants.
So I think now the field's really honing in on a scaffold within the PCM zone that consists of SPD2 and centrosomin. And what we show here in this paper is a separate scaffold that only forms an interphase and that forms between the PLP satellites and these centrosomin flares. And we think of PLP as the glue or the anchor for centrosomin in this zone, because if we get rid of PLP, then centrosomin ends up released from the centrosome and in the cytoplasm. We think that this PLP centrosomin scaffold is important because having an astral microtubule array in embryos is really important for this nuclear spacing. And so you always want to have a centrosome on, you always want to organize radial arrays of microtubules, and so you always want to maintain PCM at the centrosome.
Lerit and Rusan now want to investigate how these PCM scaffold proteins are regulated over the course of the cell cycle, and how their functions differ in different cell types. In the meantime, however, you can learn more about how PLP and centrosomin organize interphase centrosomes in the paper by Lerit et al., published in the July 6, 2015 issue of the Journal of Cell Biology.
You